Hello viewers, I am Dr. Robiul. I work as a lecturer of pathology in a medical college hospital, and I am making this video for my students and also for you. Hope someone finds this helpful. Today is our 74th upload, and today we are starting our fascinating immunology lecture series. This is the first lecture, and in this lecture we will learn what do we mean by immunity, what are the types of immunity. We will talk briefly about innate and adaptive immunity and also about active and passive immunity. We will talk about the interaction between humoral and cell-mediated immunity, and towards the end of today's lecture, we will talk about some commonly used terms in immunology and also about the organs of our immune system. Okay, a lot of topics, so let's begin. First question, what do we mean by immunity? Now, the word immune is derived from the Latin word immunis, which means exempt or protected from a liability. Always remember, in medical science, the term refers to being protected from infectious pathogens. So how can we define immunity? It can be defined as resistance to disease, specifically to infectious disease. It is the protection from infectious pathogens. So what do we mean by immune system? It is composed of collection of cells, tissues and molecules that mediate resistance to infections. And what do we mean by immune response? It is the coordinated reaction of the cells and molecules of our immune system against the various offending infectious microorganisms. And what do we mean by immunology? It is the study of immune system, including its responses to microbes and damaged tissues and its role in disease. So now that we have talked about some introductory points regarding immunity and immunology, now let's move on and talk about the classification of immunity. And as we can see, we can classify immunity into innate and adaptive immunity. Both innate and adaptive immunity can be further subdivided into humoral immunity and cell-mediated or cellular immunity. So now that we have classified immunity, now let's talk about the various types one by one. And first, let's talk about innate immunity. Always remember, it refers to the mechanisms that are ready to react to infection even before they occur. It is also known as natural immunity or native immunity. It is non-specific. It can act within minutes because it is always ready to fight against any invading microbes. However, it doesn't improve after exposure and it has no memory. So now that we have talked about innate immunity, now let's move on and talk about adaptive immunity. In case of adaptive immunity, always remember that it refers to mechanisms that are stimulated by microbes and that are capable of recognizing microbial and non-microbial substances. So adaptive immunity is occurring in response to the microbes, and it can recognize microbes as well as certain non-microbial substances, and it can act accordingly. Adaptive immunity is also called acquired or specific immunity, and as the name implies, it is highly specific. However, it requires several days before becoming fully effective. It improves after exposure and it has memory. Now in this table we are summarizing the main components of innate and adaptive immunity. As we can see, the humoral components of innate immunity will include complement and neutrophils, and the cell-mediated or cellular components of innate immunity will include macrophages and dendritic cells. However, as we will later see, macrophages and other antigen-presenting cells, for example dendritic cells, have roles in both innate and adaptive arm of the immune system. So what are the humoral components of adaptive immunity? They will include B lymphocytes and antibodies that are made by plasma cells. What are the cellular components of adaptive immunity? They will include helper T cells and cytotoxic T cells. Now in this image we can see that macrophage and other antigen presenting cells participate in both innate and adaptive arm of our immune system. So here first there is phagocytosis of microorganism by macrophage or dendritic cell, and after that there may be destruction of the microbes and secretion of cytokines, and these functions will fall into innate immunity. Again, there may be antigen presentation. The macrophage or dendritic cell may break down the antigenic components that were present in the microorganism, and then they may present those antigens to CD4 positive helper T lymphocytes, and this type of function will fall under adaptive immunity. So here we can see that macrophage and other antigen presenting cells do participate in both innate and adaptive arm of the immune system. So now we will talk about some important features of innate and adaptive immunity. As a matter of fact, we have already mentioned these features when we were talking about them previously. So regarding specificity, we already know that innate immunity is non-specific and adaptive immunity is highly specific. Regarding the time required for effect to occur after exposure, innate immunity can act within minutes. However, adaptive immunity requires several days. The immunity does not improve after exposure in case of innate immunity, and innate immunity has no memory. On the contrary, the immunity improves after exposure to microorganism in case of adaptive immunity, and adaptive immunity does have memory. So now that we have talked about innate and adaptive immunity, now let's move on and talk about the interaction between humoral and cell-mediated immunity. And in order to discuss that, you can see that I have already drawn a simple diagram in the whiteboard. So let's talk about that now. So here we can see a simple diagram that is showing functions and interactions of the major components of humoral and cell-mediated immunity. On the left side, we can see the major components of humoral immunity. Now always remember, humoral immunity is our main defense against extracellular, encapsulated, and pyogenic bacteria. So we can see that first there is B lymphocyte, and with the help of interleukin 4 and interleukin 5 that were derived from helper T cell, B lymphocyte or B cell is transformed into plasma cell, and plasma cells can produce antibodies, and those antibodies can neutralize toxins and viruses. Other components of humoral immunity, as we can see, will include complement and neutrophils, and they can act by killing bacteria and other offending agents, as we will later see. On the right side, we can see the major components of cell-mediated immunity, and they will include helper T lymphocyte and cytotoxic T lymphocyte. But as we will later see, there is also another type of T lymphocyte that is known as regulator T lymphocyte. So, helper T lymphocyte, when they are activated, they will inhibit intracellular bacteria and fungi. Similarly, when cytotoxic T lymphocytes are activated, they will kill virus-infected cells. And we will talk in details about all these mechanisms in our upcoming lectures. Now, in your textbook, you will also find these terms: active immunity and passive immunity. So now let's talk briefly about these two terms. Now, active immunity is resistance that is induced after contact with foreign antigens, for example, microorganisms. Now, contact may happen during infection. The infection may be clinical or subclinical. Contact may occur during immunization, either with live or killed infectious agents or their antigens, and contact may also occur due to exposure to microbial products, for example, exposure to toxins and toxoids. The main thing that you have to remember regarding active immunity is that here the host actively produces an immune response. So what are the advantages of active immunity? The main advantage is that it provides long-term resistance. And what is the disadvantage? It is slow in onset. Regarding passive immunity, it is resistance based on antibodies that are preformed in another host. Examples will include administration of antibodies against diphtheria, tetanus, botulism, administration of preformed antibodies to certain viruses, for example, rabies virus, hepatitis A virus, hepatitis B virus, etc. Passage of immunoglobulin G from mother to fetus during pregnancy. This is very important. Always remember this, that immunoglobulin G can pass from mother to fetus during pregnancy to provide immunity. This is an example of passive immunity. And another example is passage of immunoglobulin A from mother to newborn during breastfeeding. What is the advantage of passive immunity? The main advantage is prompt availability. Large amounts of antibody become promptly available in case of passive immunity. And what are the disadvantages? Short lifespan of these antibodies, and there is possibility of hypersensitivity reaction if antibodies from another species are used. Now this term is very interesting: passive active immunity. Here we are providing both passive as well as active immunity at the same time. It involves giving both preformed antibodies to provide immediate protection by passive immunity, and also a vaccine to provide long-term protection by active immunity. Always remember, in case of passive active immunity, the preparations must be given at different body sites to prevent neutralization of immunogens in the vaccine by the given antibodies. Examples: it is used in prevention of tetanus and hepatitis B. So here we are summarizing the main characters of active and passive immunity. So what are the mediators of active immunity? They will include antibodies and T lymphocytes. And antibodies are the only mediator for passive immunity. Regarding advantage, the main advantage of active immunity is it provides long-term resistance, and its disadvantage is slow onset. And the main advantage of passive immunity is immediate availability or prompt availability of the antibodies, and its disadvantage is short duration. So now we will talk about important components of innate immunity, and we will talk about them under two broad headings. The first one is factors that limit entry of microorganism into our body, and the second group of important components will include factors that limit growth of microorganism inside our body. So first, let's talk about the factors that limit entry of microorganism into our body. And the first factor is the keratin layer of our intact skin. And how does this act? The mode of action is it provides mechanical barrier. The next factor is surface phagocytes, and as we know, phagocytes will ingest and destroy microorganisms. Now I have a separate video entirely on phagocytosis, so you can also watch that video after finishing this video to know more about phagocytosis. The next factor is low pH in the stomach, vagina, and also fatty acids in the skin, and the low pH retards growth of microorganism. The next factor is lysozyme. They are present in tears and also in some other secretions, and lysozyme can degrade peptidoglycan. Recall that in bacterial cell wall there is peptidoglycan, and lysozyme can degrade that. The next factor that we will talk about is the respiratory cilia. We know that the respiratory tract of our body is lined by pseudo-stratified ciliated columnar epithelium, and these epithelial cells have cilia. And what is the function of cilia? They elevate microorganisms that are trapped in the mucosa, and they propel them out of our body. So that is the function of respiratory cilia, and by that function they can prevent entry of microorganism into our body. The next factor that we will talk about is defensin. They are cationic peptides, and they can create pores in the membranes of microorganism. The next factor that we will talk about is normal flora of our throat, colon, and vagina, and their mode of action is they occupy receptors, and by doing so they can prevent colonization. So now let's talk about the factors that limit growth of microorganisms that are already inside our body. So they will include neutrophils. They will ingest and destroy the microbes. Natural killer cells, they are important particularly in killing virus-infected cells. Macrophages and dendritic cells, we have already talked about their function. They can ingest and destroy microbes, and they can also present antigen to T helper cells to initiate adaptive immunity. Interferons, they will act by inhibiting viral replication, and complement. Among complement, the complement 3B will act as opsonin, and membrane attack complex, that is composed of complement 5B to 9, that will act by creating holes in the bacterial membrane. Transferrin and lactoferrin, they will sequester iron that is required for growth of the bacteria, and by doing so they will also limit growth of bacteria. Inflammatory response are also important here. They will prevent growth of microorganisms by limiting their spread. Fever is another protective mechanism, and by elevating temperature, fever can also retard bacterial growth. And the last point is very important as well. apolipoprotein B RNA editing enzyme, they will cause hypermutation in retroviral DNA and mRNA. So now let's talk about the components of innate immunity, and first we will talk about the epithelia. We know that epithelia provide mechanical barriers to entry of microorganism from external environment, and they also produce defensin, and lymphocytes that are located in the epithelia also combat microorganisms at these sites. The next cell that we will talk about is monocyte, and also we will talk about neutrophils. Now, they are phagocytes in the blood. They can be rapidly recruited to the site of infection. And the last bullet point is very important. Monocytes that enter the tissue and mature are also known as macrophages. Regarding dendritic cells, always remember that these are specialized population of cells. They are found in epithelia, lymphoid organs, and also in most tissues, and they can capture protein antigens and present peptides for recognition by T lymphocytes. They have rich collection of receptors that can sense microorganisms, damaged cells, and stimulate secretion of cytokines. Regarding natural killer cells, always remember that they provide early protection against many viruses and intracellular bacteria. Now, this term is very interesting, innate lymphoid cells. Now, these are cells with appearance of lymphocytes. They will look like lymphocytes, however, they will have features like cells of the innate immunity. Now, other components of innate immunity will include soluble proteins like complement system, mannose binding lectin, C-reactive protein, etc. And always remember, lung surfactant is also a component of innate immunity. So now that we have talked about the components of innate immunity, now let's move on and talk about the components of adaptive immunity. And lymphocytes and their products, including antibodies, are the components of adaptive immunity. And here, highly diverse receptors are used to recognize a vast array of foreign substances. So what are the cells of adaptive immune system? We have already mentioned T and B lymphocyte. Now always remember, T and B lymphocytes are morphologically quite similar. However, they are remarkably different and specialized in their molecular properties and functions. Lymphocytes and other cells involved in immune responses are constantly circulating among lymphoid and other tissues via blood and lymphatic circulation, and this promotes immune surveillance. Now, in your textbook, you will also find these terms: naive lymphocytes and effector cells and memory cells. So what do we mean by naive lymphocytes? These are mature lymphocytes that have not yet encountered the specific antigen for which they have specific receptor. So they are called naive lymphocytes. They are also known as immunologically inexperienced lymphocytes. Now, what do we mean by effector cells and memory cells? After activation by recognition of specific antigen and other signals, the mature naive lymphocytes will differentiate into effector cells and memory cells. Effector cells will perform function of eliminating the microbes, and memory cells, they will live in a state of raised awareness, and in case the microorganism return, the memory cells can react rapidly and strongly to combat the microbes. So regarding the lymphocytes, always remember that there are three populations of T lymphocytes. They will include helper T lymphocyte, cytotoxic T lymphocyte, and regulatory T lymphocyte. So let's talk about their function now. The helper T lymphocyte or helper T cell can stimulate B lymphocytes to produce antibodies. They can also activate other leukocytes and phagocytes to destroy microbes. Cytotoxic T lymphocytes, their function is to kill infected cells, and regulatory T lymphocyte, they limit immune response, and they also prevent reaction against self-antigen. So now that we have talked about the various components of immunity, now let's move on and talk about some commonly used terms in immunology. The first one that we will talk about is antigen. These are molecules that can react with antibodies. We can also define antigen as a substance which can stimulate antibody production when introduced into the tissues. Usually they are protein in nature. What do we mean by immunogen? These are molecules that induce an immune response. Now always remember, in most cases antigens are immunogens and the two terms are used interchangeably. However, there are some exceptions, for example in case of haptens. So what do we mean by hapten? These are non-protein substances. They have no immunogenic property by themselves. However, when they are combining with protein, it can form a new antigen that is capable of forming antibodies. What do we mean by antibody? It is a protein substance that is produced as a result of antigenic stimulation. Circulating antibodies are immunoglobulins, and there are 5 classes of immunoglobulins, and they will include immunoglobulin G, A, M, E, and D. You can remember them by thinking about GAMED. The last topic that we will talk today briefly is regarding the structure of immune system. Particularly, we will talk about the primary and secondary lymphoid organs. Always remember that primary lymphoid organs will include the thymus and bone marrow, and secondary lymphoid organs will include lymph nodes, spleen, mucosa associated lymphoid tissue that is present in respiratory tract, gastrointestinal tract, etc. So always remember these organs for your examination. So this concludes our first lecture of immunology. I hope this video was helpful. If you like my videos, do comment, share, subscribe, and let me know. And for my students, I will also recommend you to go through your textbooks to know more. Okay, that's all for today. Until next time, take care and stay blessed. Thank you.